At least 99.9% of the bacteria that are out there are harmless 99.9% of the time. And it's probably even less than that. And it's such a tricky, really important question. How do we identify and do something about that less than a tenth of a percent of the time that's going to cause trouble? I'm a research microbiologist at the US Meat Animal Research Center, and it's primarily focused on pathogens and commensals in the beef and pork production continuums. That means all the way from birth through feeding, through the end stages of feeding, through processing, all the way to the final product, but also the byproducts, including manure. The research is important because bacteria have such a broad impact. They shape our soil fertility, they shape animal and human health. And while most bacteria can be classified as commensal and won't cause an issue, a very small percentage of them can be and could become pathogens. And we need to increase our understanding of where these bacteria inhabit, what influences them, so we can improve global health, really One Health, the environment, animals, and people. This is a plate where the black colonies on here, if you can see them, are salmonella. This is how we would detect salmonella by the most sensitive method we have right now, but also the individual colonies. We would pick one of these, and actually we would streak it on these plates to further purify it so we know the individual colony, or what we call isolate. And then with that, that's what we would whole genome sequence or target specific genes or perform some type of other assay in the lab. But the first step after you detected it is to get it as an isolate. The same antibiotics that you or animals are exposed to into this media, so we get the specific resistant salmonella or E. coli. So there's a lot of different types of resistance out there, and we've only really scratched the surface of understanding it. So it's really important to define how each of those antibiotic uses impact the bacterial populations. Antibiotic resistance has been identified as a major global concern. And the problem with this is antibiotic resistance is an intrinsic part of bacteria. Any place where you have bacteria, you will find antibiotic resistance. The question is how much do our antibiotic uses contribute to that antibiotic resistance? Do you increase it or change it? Beef production is so important to Nebraska and the US economy, and everybody wants safe food. And anything we can do to increase the safety of food is a societal benefit.